What happened to the whiteboard? Can you see the board? Yeah.
Yes, sir.
Hello? Yeah, so the recording is on now. Yeah, so the real point is that we talk about IGEM conference, and these are some things that people should participate in. It's a very useful part where each of these plasmids, etc., we call them as building blocks, and we use a Lego kind of strategy to make all kinds of very interesting structures, and you can use your creativity to its fullest extent. And this is a conference I remember in 2000, I was in Cambridge, and that was the year it was started. I suppose many of you had just been born at that time, or not yet. They were telling me that if IIT or whichever place participates from India, you will definitely win the first prize, because there are 0 participants. Of course, now it has become much more competitive. So that's an option that we missed out. But yes, IGEM has become very popular over a period of time, and International Genetic Engineering Machines. And you can participate, go to the US, present your interesting, clever ideas, and also learn how to use biological systems as Lego blocks to build all kinds of very creative things, including electronic circuits which are mimicked by biological systems and so on and so forth. So this is something which I leave for your creativity and imagination. This is something which I have found is very appealing and very interesting to most students, IGEM. So think about it, talk to somebody, and if you want to participate, maybe some faculty or the other will be able to help out. I did this long time ago, 15, 20 years ago. So now you take this gene and you insert this, and this is the gene of interest. This is the gene of interest which is inserted, and this gene of interest is nothing but this gene is going to make your protein of interest. So you can insert it, but normally PUC19 is what is called a cloning vector. The reason it is called a cloning vector is that it can clone genes, but usually these genes do not necessarily produce the protein. And why is that? Because to have the gene of interest producing the protein, you need to have a promoter and a ribosomal binding site and so on and so forth, plus a ribosomal binding site, because the promoter will make the mRNA. So as I said, remember your original logic, DNA will give rise to RNA, or mRNA will give rise to protein. Now these things will not happen automatically. So the DNA will do nothing, and if you put a promoter, you will get your mRNA, because the RNA is made. Whether the RNA will be translated, this is transcription, and whether it will be translated to the protein of interest will depend on the ribosomal binding site. So expression vectors are different from cloning vectors, and expression vectors contain all these other things. So they are also plasmids. And so only thing is that they contain the appropriate upstream sequences which can transcribe and translate the DNA to RNA and then onto protein. And this is where what is usually taught in recombinant DNA technology. And they say, well, very easy job. Take the gene of interest, plug it into an expression vector downstream of the promoter, and your job is done. You can make your protein. Now, as I would like to sort of teach you today and in the next few classes, the job of the engineer begins where the job of the biologist ends, which is to say that once you have inserted this plasmid, and then of course you take this plasmid and put it back inside the cell. This cell obviously does not contain any plasmid. So when you transform this cell, you can select the desired cells on the basis of antibiotic resistance. And if the antibiotic resistance is present, then of course the gene is also present, and you can then use that cell for production of the protein of interest. There are various ways to sort of do this. For example, earlier, have you heard of blue-white screening? Has somebody mentioned it in the last class? Blue-white screening?
Yes, sir.
No. Yeah,
Yes, sir, we have.
You have done this, right?
Yes, sir, we have.
So blue-white screening is again essentially disrupting the lag-Z and introducing the gene of interest in the middle of lag-Z. And so the removal of lag-Z prevents the beta-galactosidase from being produced and hence not being able to degrade the X-gal, I'm going through this very fast because these are things that you have done. And so you get white colonies. It's proof that the gene has been disrupted. It cannot produce beta-galactosidase, and the gene has possibly been disrupted by your gene of interest. And your gene of interest is the one which is what you're trying to produce. And if this gene of interest is downstream of the desired promoter, then your job is done. So really all the things that have been taught to you are in terms of cloning. But right now we are interested in expression. Because it's not just what we want to clone, but we want to express. So therefore blue-white screening and all that is usually not preferred, because we would like not to have any laxity or any useless stuff around the plasmid. We just want the appropriate promoter and the RBS and the gene of interest downstream. Of course, the origin to retain the plasmid, the ampicillin resistant to select for the plasmid, and the gene of interest to produce the protein of interest. And this is then transformed back into the cell, inserted back into the cell, and then we can make our protein of interest, or so we think. But many times this production does not happen. So there are two basic issues that we will talk about. One, why is it that proteins are not always produced? What are the issues proteins not produced? And the second, and equally important as engineers, which is that how do you scale up this process? Because finally you are interested not just producing it in a small scale in shake flask as a biologist does, but to scale it up to larger scale production. And what are the issues that come up when you do larger scale production of recombinant proteins? And why is it that proteins are not produced, and what are the solutions available to that? These are the two questions that we will try to address in the next few classes, over and above what is usually taught or what will be taught to you in recombinant DNA process. So this is what is a very, very short and brief overview of what biologists think is cloning and expression all about. I mean, take the gene, bung it into a expression vector, job done. Now, over a period of time, the promoter that is used has changed. The promoters, remember that promoters, the stronger the promoter, the better it binds with RNA polymerase, the more efficiently it transcribes. Now, remember that the story that we were talking about, DNA to RNA to protein. Now, this is an oversimplified picture, but it's also sometimes important. Now, the assumption, and then folding, and then export, and so on and so forth. Now, there is an assumption by biologists that the first step, this step, the transcription step, is the rate controlling step. As biologists, they think that this step, transcription, is rate controlling. This may or may not be true, right? The first step is usually rate controlling. When you have put in a promoter and you're trying to convert DNA to RNA, you can think that transcription may be rate controlling, but this is a question mark. It may or may not be true. So people have thought that as they make stronger and stronger promoters and make more and more RNA from the same amount of DNA, we might actually end up getting a large amount of RNA and hence a large amount of protein. Now, this assumption is only true if transcription were to be rate controlling, which is not always true. So there has been a series of studies where the promoters, the earliest of promoters, the most common promoter used to be the lac promoter. You have heard of lac, the lac paradigm. Lac is an inducible promoter in the presence of lactose. It gets yeah, but lactose would tend to get consumed, so the promoter would no longer remain active. So usually in industry, we use something called a IPTG, which is a, what is IPTG? Any one of you?
Sir, IPTG is also used in lactose promoter, I guess, like it is a reagent and it is a chemical reagent, I think.
It is a chemical reagent. That is very obvious. It cannot be otherwise. Isopropyl thiogalactopyronoside. But what is its property? It mimics lactose in terms of
yes, sir, and yeah,
it mimics lactose, but it does not get degraded like lactose. Okay, so this is also an inducer, And I want you to think about what is the name used for such inducers, gratuitous inducers, which mimic the regulatory property of the molecule but do not belong to the same molecule. What are these people, these such chemicals called? Think about it. Every question will not be answered. Okay, so IPTG is used to replace lactose because unlike lactose, it does not get consumed. So the induction is continuous. So IPTG is a better inducer than lactose because of its being not consumed. And so we can add it in very small amounts, 1 millimolar, half a millimolar, and that is good enough for inducing lactose. Now, instead of lactose, some people started using trip for the tryptophan operon. And then, of course, people have moved to combination of lactose and trip, which is called a TAC promoter, again induced by IPTG. So these are from lac to trip to TAC, promoters become stronger. And today we have more than 100 promoters available to us with varying strengths. And people have very strong promoters nowadays. One of the most commonly used strong promoters is a promoter called the T7 promoter. Have you heard of T7? T7 is a, yes.
Is it a promoter of T7 phage
yes.
virus?
Yes, it is a promoter of the T7 phage virus. So now someone of you should ask this very question: how the hell can I use a T7 promoter in phage? Where is the cognate RNA polymerase? Because T7 promoter will recognize the T7 RNA polymerase, right? So we have a trouble. And so where is the T7 RNA polymerase, which will be required for activating this DNA? It is a very strong promoter, but of course there's this very minor problem that you do not have the T7 cognate T7 RNA polymerase. So now what people have done is they have taken E. coli and in the chromosome they have inserted the T7 RNA polymerase. And this T7 RNA polymerase is inserted downstream of the lac promoter. With lac promoter and T7 RNA polymerase is inserted, and these strains have the label. They are introduced. This T7 RNA polymerase along with lac is introduced by a mechanism called lambda phage. So it is introduced. So therefore these strains, for example, if this original strain name is BL21, then this introduction is done by a lambda DE3 phage. So,
sir, I have a question.
Yes, please.
Sir, you were saying that T7 RNA polymerase is inserted including this lac promoter. Sir, T7 RNA polymerase is a enzyme, right? And lac lac operon you were talking about, that is operon in the DNA. So, sir, how these two are inserted at one place? I am not understanding.
Okay. So whenever I say, whenever I give a name that T7 RNA polymerase is inserted, by definition, you have to remember that it is not the enzyme. Enzymes cannot be inserted in DNA, right? It is the gene for, this is a shortcut language. Whenever I say I have inserted T7 RNA polymerase or I have inserted this protein, you obviously do not inserted a protein. You have inserted the gene for the protein. So this is a shorthand language that you should excuse me and everybody uses. It is assumed that when I say T7 RNA polymerase, I mean the gene for T7 RNA polymerase. And the lambda promoter is picked up, the lac promoter, just the promoter region and the RBS region is picked up and attached upstream of this T7 RNA polymerase. This is the T7 RNA polymerase gene, and this is the lambda, and this is the lac promoter. The lac promoter and the T7 RNA polymerase gene are attached together, they are joined together, and this gene is taken and inserted here. Okay, so this lac and this T7 gene are this whole combination of this whole genetic element containing the lac promoter and the T7 RNA polymerase are inserted into the chromosome of E. coli using lambda DE3, and therefore many of these strains are given the name BL21. For example, is the standard strain used of E. coli. You will see E. coli BL21 lambda DE3 or DE3. Sometimes the word lambda is missing. At BL21 DE3. which really means that the chromosome of this E. coli strain contains, using the lambda phage, a insert of T7 RNA polymerase with the LAC promoter. So when you add IPTG to this, what it will do is the LAC will get activated and it will produce a certain amount of T7 RNA polymerase. Now, parallelly, you have the plasmids inside. And each of these plasmids has the T7 promoter. The T7 promoter. And each of these T7 promoters has downstream of the T7 promoter the gene of interest. Your gene of interest. The gene that you want to make. The gene of interest is downstream of this T7 promoter. So now you see it's a multi-step process. You added IPTG, activates the LAC, activates the T7 RNA polymerase. This T7 RNA polymerase goes and binds with the T7 promoter, which is present in the plasmid, which is inside the cells here. And these plasmids contain the T7 promoter here. And these T7 promoters then get activated and produce, or rather transcribe, the gene of interest. And therefore, you can make a very large amount of protein based on these systems. And this is something which in the practical classes, my students will show to you in the lab. Of course, this will be online. Next week, you should see or ask about the T7 promoter and expressions based on T7 promoter and how we use this cell, the BL21DE3 cells, to use it with the T7 promoter. And these are different plasmids. They are usually called PET plasmids or PR set plasmids. PET22B will be the name of a plasmid. Various names. My students will tell you what are the special features of these things. And these plasmids are then used to express the protein of interest under the T7 promoter. And the answer why T7 works is because the host cell has the T7 RNA polymerase in the chromosome, which is then, so the induction is not of the T7. The induction is of the T7 RNA polymerase. So don't think IPTG is inducing the promoter here. The IPTG is actually inducing the promoter in the chromosome and allowing for production of T7. So a little amount of T7 RNA polymerase then makes a very large amount of your gene of interest, or transcribes the gene of interest, because of the very powerful nature of the T7 promoter. Is this clear? Because one copy of T7 RNA polymerase will make multiple copies of your gene. I mean, it gets reset. Yeah. Okay. So this is how it works. And so you can see this is how very large amounts of expression is obtained. Now, let us get back. These are very basic molecular biology concepts, and I'm sort of rushing through them because you will hear about them in many courses. Let us get down to the more engineering and more interesting issues that are not usually addressed in the usual course of teaching. Now, when I am growing cells in a bioreactor, cells are going to double. So when I'm growing cells in a bioreactor, cells are going to double. And of course, one cell will become two, let us put it very crudely, and two cells will become four, and so on and so forth. Right? And we can think of more and more and more cells. Now, remember that this fellow here has plasmids. And this partitioning of plasmids, has to be, some plasmids will go here, some plasmids will go here. And this plasmid partitioning need not be exactly equal. Especially for high copy number plasmids, which we are talking about, high copy number plasmids, this partitioning is what is called random. So plasmids partition into daughter cells in a random fashion, right? And this randomness, usually by the time it divides again, then this copy number is corrected, but it may or may not undergo full correction. So it is well possible that the next round, this may have nothing. So you can have a distinct probability of plasmid-free daughter cell emerging.
Sir,
in the course of cell division. Yes.
Sir, I have a question like when the first cell is dividing and it is becoming two cells. In both the cells, there will not be equal number of plasmids. It's good. This is fine. But next comes like the plasmids will replicate in that cell also, and it will become like there are regulators. I guess which maintain which are responsible for maintaining a significant number of plasmid in the cell. Like if it will be less than that, they will make that number of plasmid and maintain that number of plasmid. So how can it be like on the third stage, like in the second generation, there will be some cells in which there won't be any plasmid?
हाँ, मैं बता रहा हूँ, ये एक स्कीमेटिक है. आप ये मत सोचिए कि सेकंड जनरेशन में ऐसा होगा. हो सकता है थर्ड में हो सकता है फोर्थ में हो सकता है 10th में हो सकता है कभी भी ना हो. So let us do the arithmetic behind it. First of all, this was for illustration only. This is not at all an attempt to tell you that आप लोग second generation में ऐसा होने वाला है. Now you have raised a very interesting question, and I'm not going to answer it very clearly, deliberately, because it becomes into very complicated biology. Now, plasmid replication, plasmid partitioning is random. Now, which really means that that if there are n plasmids, then the ways in which it can distribute into two daughter cells is given by the binomial distribution. Right? You are familiar with the binomial distribution? So the binomial distribution is what will determine the copy number distribution in the daughter cells. In any, remember, this is not one cell, this is 1 million cells or 1 billion cells. So 1 million cells are dividing into 2 million and 2 million into 20 million and so on and so forth. You know, so don't think of this. This is a simplistic imagery that I'm drawing for you. The real picture is much more complex. Now, a binomial distribution takes place. Now, there are two questions that I'm going to answer for you. One, if n is the copy number, given the binomial distribution, the probability that even with n cells, one cell will have 0 and everything else will get all of them will be given by the probability of plasmid free cell emerging with n copy number, plasmid free cell emerging. This probability will be given by 2 to the power of 1 minus n, right? Because what is the, or let me put it like this, 1 upon, see, okay, let us do it very simply so that, you know, you may be, most of you should be familiar with statistics, but I always feel that, see, I have coins with me, right? I have n coins. I think people are familiar with this. N coins with me. What is the probability that all heads will happen? That is, all n coins will all turn out heads. The probability of one head turning out is half, and into half second head, and till n. So 1 by 2 to the power of n is the probability that all heads will come. Is this all right?
Yes, sir.
And the probability that all tails will come is also 1 by 2 to the power of n, all tails.
Yes, sir.
Now, really, what it means, I'm saying, now this is the level at which I do not want to teach BTech students, but yes, okay, fine. For once, all heads means the plasmid has gone all to this fellow. And all tails is that the plasmids have all gone to this fellow. So it is only when plasmid goes all to this or all to this. So 1 by 2 to the power of n plus 1 by 2 to the power of n is the probability that one of the daughters becomes plasmid free. So the probability of plasmid free cell emergence is 1 by 2 to the power of n into 2, which is 2 to the power of 1 minus n. Okay, so the probability of plasmid free cell emergence is 2 to the power of 1 minus n, which is straight away available from the binomial distribution. I sort of tried to make it very simple. Now, the probability of plasmid free cell emergence, if n is the copy number, is given by 2 to the power of 1 minus n, where n is the copy number. Now, there are two issues here that you need to remember. One, that n is the copy number, but n is what we measure, n is the average copy number. But you could have fluctuations in copy number, which is what you were asking, Ratnesh, and I'm going to give you the answer now. Now, suppose I had 50 copy number in the beginning, and when it divided, some cells would get 5 and 45. I mean, binomial distribution, some probability, or 10 and 40. I don't really care. I'm just giving you numbers for your appreciation, or 3 and 47. Now, what happens is that the copy number has a corrective mechanism. And that is what I've said. I don't want to get into too much complicated biology. When the copy number is already 47, the cell is able to see that there are 47 origins. I talked about ORI. So when we have, so the origin of replications. So this is the cell, this is the plasmid. And remember that this is the origin of replication, which allows plasmids to replicate independently inside the cell. Now, the cell is able, the host cell is able to interact with this origin and say, well, you have already 47, so it does not allow too much of division, and 47 over a next period of time will become 50. 3, on the other hand, may not be able to become 50 by the time the cell divides again. Why? Because 3 will become, by the time, usually plasmids divide much faster than the cell. And therefore, there is a autocorrect mechanism which you were talking about. Yes. So by the time the cell divides, 3 might undergo 3 divisions, 3 to 6, 6 to 12, and 12 to 24. Still, it has not completely corrected because the cell has inherited very few plasmids. The ability to autocorrect. The ability to, now let me just introduce some funny complications deliberately. The ability to autocorrect also depends on the length of the gene that you have inserted in the plasmid. Usually plasmids autocorrect very fast, but when you have inserted genes of your own interest, and if these genes are large, the autocorrection ability becomes compromised. So the ability to autocorrect becomes less and less as you introduce more and more complicated genes. But you have to introduce complicated genes. It is your job. You're not bothered about the cell being able to autocorrect or not. You are interested in the cell producing your product, right? You're not going to go into all those details. So sometimes when we introduce large genes into plasmids, the ability to autocorrect becomes less. So every cycle, the 50 copy number that you think the cell has tends to become, this copy number distribution becomes skewed. And the moment the copy number distribution becomes skewed, now I hope you understand the maths of it. Then the copy number distribution, these are the things that biologists don't tell you. So that is why we want to do about this. When this is a skewed distribution, then remember that the average copy number is 50. But some of these are 60 and some of these are 3 and 5. And 3 and 5, and this 3 and 5 copy number and 3 and 5 and 10 copy number are going to contribute significantly high to the average probability of plasmid free cell emergence, because the probability of plasmid free cell emergence is given by 2 raised to 1 minus n. So remember that if the copy number is 50, the probability of plasmid free cell emergence is P is equal to 2 raised to 1 minus 50. Which is 2 raised to minus 49, which is a very small number. And the average copy number is 25, still P is equal to 2 raised to 1 minus 25, which is equal to 2 raised to minus 24, which is still a very small number. But if the 50 copy number is a mix of copy numbers of 1, 2, 3, 4 until 50 in a skewed sense, then this small population which has a copy number of 3 and 4 and 5 will add to probabilities of 2 raised to 1 minus 5, if 5 is the copy number, or 1 of 2 raised to 1 minus 3, if 3 is the copy number. And these numbers are very large. This is, 25%, 1 minus 2, right? So these cells might be small in number, but they have a very high probability of plasmid free cell emergence. So the sum total of probabilities over a population of plasmid free cell emergence is significantly higher than what you would if you had this very simplistic notion of the probability being 2 raised to the 1 minus 50 and hence very small, and hence we don't need to bother about it. We have to bother about it because of the copy number distribution. And so this is one issue of copy number. The second problem, which is adding to the more complex problem, remember that I asked this funny question, which maybe you should answer. Suppose I have 50 coins and I have a bet with you. If I get 50 heads or 50 tails, then you will give me 1,000 rupees. If I get any other combination, 10 heads, 40 tails, 25 heads, 25 tails, all other combinations, I will give you 1,000 rupees. So will you take the bet? Most of you will take the bet. We'll say, well, because the probability of getting 50 heads and 50 tails is when Professor Mukherjee will take 1,000 rupees, and when it is any other possibility. But then I will perform the experiment my own way. And how will I perform it? I will take the coins and I will stick them together. I have joined all the 50 coins together. If I join all the 50 coins together, then it will all be 50 heads or 50 tails. And so I get 1,000 bucks. Now, which really means that if the coins are joined together, then they act as one unit and not as 50 units. So then this probability does not work. So n is the copy number. But n has two meanings. The cell counts the number of origins as the copy number. Number of origins. But plasmids have a bad habit of undergoing something called multimerization. Which means that plasmids can form chains like this. So this is a former or a dimer. Or dimers can be like this, dimers can be a much larger plasmid like this. Or a trimer or a multimer. So each of this 4 plasmids has 4 origins. So this will be treated as copy number of 4 by the cell. So it will not allow you to divide. But for purposes of inheritance of plasmid or division into daughter cells, this is only 1 unit. So the effective n, the n effective is significantly lower than the actual copy number of the plasmid. So you think it is 50, but the number of discrete units after multimerization may only be 25. If say all of them have formed dimers. So all these tend to increase the probability of plasmid free cell emergence significantly in the culture as cell division takes place. Is this argument clear to you, all of you?
Yes, sir.
Let me stop here. Yeah. So the plasmid free cell emergence is a function of probability. This probability is determined by various factors, but the story is not yet complete. Now that is one thing. Now let us think about the other aspect, when cells are grown. Now, here is a cell and the cell is growing and it contains some plasmids, and for some reason or the other, when it has divided into 2 cells, some way, this is just for illustration purposes, this has inherited all the plasmids and this fellow, poor fellow, has no plasmids. Now this is a plasmid free cell, and the emergence of this plasmid free cells is associated with a probability P of emergence. This probability could be very small, 1 in a million or 1 in 10 million. Because, 2 raised to 1 minus 50 is very small, or 2 raised to 1 minus 25 is very small. So, and so on. So let us say the P is equal to 10 raised to minus 5 or 10 raised to minus 6. Something like that. Let us say. Now, we have another problem. And the problem is that I am growing this cell. So this cell is going to take this cell is now containing my gene of interest. And this gene of interest is being used to make the protein. Now this cell is producing protein. This cell is not producing any protein. Now remember, protein production utilizes cellular resources. And if you are utilizing cellular resources, you are going to end up with diverting cellular resources for the protein that you want. Cell does not want it. You are forcing it to make the protein. The cell is not interested in producing the protein. This is the protein that you want. And so cellular resources are being diverted to what you want. So what do you get? You will get a decline in growth rate, a metabolic burden, a decline in mu. I hope you know specific growth rate, mu. So metabolic resources are being diverted, and this metabolic resources leads metabolic resources diversion leads to a what is called a metabolic burden. A metabolic burden. This metabolic burden will get translates to lower growth rates. So, without too much of details, let me straight away jump into the effect of lower growth rates. Now, let us do a simple equation. Let us say that I define alpha as mu minus over mu plus. Do you know what this is? Can you tell me what I'm trying to do? So alpha is the measure, is the growth rate of plasmid free cells denoted by minus divided by minus plasmid containing cells. So alpha is always greater than 1. Are you getting this argument? Because of metabolic burden, the cells, the plasmid free cell will always grow faster than plasmid containing cell.
Yes, sir.
So there are two things happening. One is there is a very small probability of plasmid free cells emerging. But once they emerge in the culture, they are able to grow faster. So given sufficient time, they will always overtake the culture because alpha is greater than 1. So you may think that your situation is safe because your probabilities are low. That's 1 in a billion or 1 in a million. But this 1 in a million cell that emerges has the ability to grow faster. And the relative level at which it grows faster depends on the relative level of this burden. And that depends in turn on the amount of protein that you want to make. But your industry, as an industry person, you are trying to maximize protein production because you want to maximize your product. And the more you maximize your product, the more burden you increase, the more you increase the growth rate differential, alpha. Sorry. So the alpha increases more and more because your desire is to make more product. So you have this problem that plasmid free cells emerging will then be overtaken very fast the more efficient your product formation methodology is. And in the end of the fermentation run, you can end up with cells which are not producing at all because they have been overtaken by plasmid free cells. So this is the problem of scale up. So when you want to grow it in shake flask, now I will explain this. No, it's not very obvious. Let me see. So let me write the equations. You see, let us write it in a CSTR. Let us write it in a CSTR. It's easy to understand. Take a CSTR. Consider a CSTR. You're all familiar with this, right? So I can write now a material balance, a biomass balance on both the plus cells, the plasmid containing cells, and the minus cells. So let us consider that's accumulation V dx by dt. Are you familiar with all this, or am I going too fast? Is equal to mu plus x plus minus dx plus. आप लोग ये familiar है आप लोग? or am
Yes,
I
sir.
writing?
Yes.
ये चलेगा ना? हाँ. And this is mu plus x plus. This is the rate of, this is accumulation. This will be steady state accumulation. This is growth. This is outflow. मैं assume कर रहा हूं कि आप लोग जानते. This minus P, the probability into mu plus x plus, where P is the probability of plasmid free cell emergence, and this is the number of cells which get converted to plasmid free cells. So mu plus x plus is the rate of growth, and as it is growing, a certain fraction of this, a very, very small fraction, say 1 in a million, is getting converted to plasmid free cells. So that is becoming plasmid free here. This will be written here. So notice that the second equation that I will write will be V dx minus by dt is equal to mu minus x minus dx minus plus P, notice, mu plus x plus. This is the amount that gets converted. This is essentially what is happening because of plasmid instability. This number may be very small. So this is the material balance for plus cells, and this is the material balance for minus cells. Does that make sense? Does these two equations make sense to you?
Yes, sir.
Because you should point out, otherwise, we will go slowly. Some amount of engineering is. So this is growth. Now notice that mu minus is greater than mu plus. And I have said alpha is equal to mu minus upon mu plus. Now notice that, and the initial condition, is this is x plus is 100%. And x minus is 0% in the beginning. It is 0. So slowly a certain amount of fraction will start getting converted into this, and this then will grow on a faster rate. Are you getting this argument? So I will sort of try to point out that I can solve this equation, but I will not solve it here. But it is slightly irritating, and then we can plot this versus time. And what we can plot is, we can plot x plus divided by x plus plus x minus. Or rather, we can plot percentage plasmid containing or percentage recombinants. X plus divided by x plus plus x minus. And in the beginning, of course, the starting point is this is 100%. So very small probability of plasmid free cell emergence. And what is going to happen? This plasmid free cells are going to emerge very, very slowly. So for a very long time, this will remain 100%. And after a very long time, this will become 99.999%. And after a very long time, it will become 99.99%. And after a very long time, it will become 99.999%. And after a very long time, it will become 99%. Are you getting the point? Because the plasmid free cells are going from 0.0001% to 0.001% to 0.01% to 0.1% to 1% to 10% to 100%. Are you getting this argument? Because it's exponential. Because mu minus is greater than mu plus. So it's the inverse exponential function. So for a long time, you will see the reactor has having only 100% of your cells, but it's not actually 100%. It's actually 99.99999. And after some time, and this is 0.00001%. And after some time, this becomes 0.001%, and still you can't see it, and it becomes 0.01%, you can't see it. 0.1% you might just about able to see it, and then 1%, and then 10%, then 100%. So suddenly, overnight, your cell suddenly becomes plasmid free. Now, the rate at which it overtakes, so I will so really what I'm trying to point out here is that if I were to plot time or number of generations, time or number of generations. Both of these are the same thing, right? Number of generations elapsed in the reactor. And I were to plot percentage recombinants, then I would have a graph like this. Where this long length represents a low value of P. If P were even lower, then my graph would be it emerge even later. If it was even, so this may be a P equal to 10 raised to the -5. This may be a P equal to 10 raised to the -6. This may be a P equal to 10 raised to the -7, and so on and so forth. While the alpha determines the rate at which it declines. So if alpha were equal to 1.4, maybe this is the graph that you get. But if alpha were equal to 1.3, maybe this graph would be slower. Or maybe if alpha were equal to 1.1, maybe this decline would be even slower. So once it emerges, so there are two things here. Emergence of plasmid free cells, which is determined by P, and overtaking by the plasmid free cells, which is determined by alpha. So there are two phenomena. It emerges and it overtakes. They are related actually. I'm just for conceptual clarity, I'm making this two different. So emergence of plasmid free cells is happening because of the probability of emergence, which values are very low, as I told you. But depending on the metabolic load, your plasmid free cells may be lost very, very quickly or slowly in the culture. So you can see that this whole yes, go ahead.
I have a question. Like, does it really happen like that? All the high number of plasmid containing cells are vanished after some time, or they become like really low percentage?
Yes.
Like
it disappears. हाँ ये होता है और उसके solutions हम बाद में बताएंगे. I want to tell you the problem first, and there are solutions to it. I will talk about the solutions in the next class. It does happen. Plasmids can disappear completely from the culture and in a large scale culture and leave you with nothing.
Sir, in that case, how does this high number of plasmid containing cells exist in the nature?
Well, they did not actually, because they existed not as chimerics. Notice that the metabolic burden that I introduced, you are modifying the plasmid and introducing your gene of interest. You are disrupting the natural plasmid host cell synergy or coexistence by actually using the plasmid to make your own product of interest. If you were to look at the natural original plasmid, that was able to stay. So we are not talking of the stability of natural plasmids. We are talking of the stability of non-natural plasmids, which are the vectors that we have made for our production purposes. Though the question that you asked was a question that I worked on many years ago and used it to develop some very interesting technology, but I think that would be more of research subject. It is a subject that I don't want to. I can talk about it in the class if you are interested, but I don't know how many of you are. We'll be going into too much of depth in the subject. I can talk about it. This question as to then how do plasmids naturally stay? And that question gave us a very good answer of a very wonderful idea of a technology that we developed called the Q-cell technology. Maybe I'll mention it in 5 minutes in the next class. So is this point clear? Let us have the concept. Is the concept clear? Plasmid free cells emerge and they overtake.
Yes, sir. Yes.
The emergence is due to probability. The overtaking is due to alpha. So let us stop here. Yeah. Any more questions?
I had a doubt that we are designing for culture metabolites that we are feeding them in substrate to optimize the production of protein. Okay.
Yes.
So in that case, if a plasmid cell free emerge, so its
Yes.
growth should be less than now, means mu minus should be less than mu plus. Why it is greater? Means
Why will it be less? I mean, I didn't get your argument. Why should it be less? The mu
because we are
minus
designing
in,
our metabolites to such that the only cells which are generating plasmids can consume much faster that substrate.
ऐसा कुछ नहीं होता है. ये ऐसा मीडिया आज तक डिजाइन नहीं किया गया. आई एम सो सॉरी टू sort of disappoint you. The media that is used for growing cells remains the same, which is yeast extract, peptone, sugar, glucose, magnesium. So there is no way that you can design a media such that it is consumed only by cells which produce the protein. If such a media could be designed, life would be very simple, but such a media does not exist, and it cannot be designed. Theoretical considerations also, because the cell consumes food for its own growth, and the plasmid
every cell has same consumption rate, whether it's plasmid free or plasmid containing.
As a matter of fact, it could be argued that cells which are containing the plasmid and producing the product would have a more inefficient way of utilizing substrate and less efficient way of utilizing substrate, because their system is disrupted. They are no longer evolutionarily as fit as the original wild type strain.
Okay.
You understand?
Yes, sir.
So as a matter of fact, they are less fit, and so their ability to eat food is compromised, but they eat the same kind of food. Food is food. Carbohydrates, proteins, lipids, etc., etc. Vitamins, yes. Whatever. Yeah.
Sir, I have another question. Like, in the beginning, how do we sort out, like, how do we get high number of plasmid containing cells?
Because we select them on antibiotic. We transform them on a plate. We introduce the plasmid inside the cell, and when we transform them in a plate, we select it on a plate which contains antibiotics. So only those cells which have high copy number of plasmid are able to resist the presence of antibiotic and grow. So the individual colony which emerges on the plate is full of high copy number plasmids. So then obviously the question that you should ask is that why does the antibiotic not take care of plasmid free cells? It should kill the plasmid free cells. And yes, the answer is true, that one of the ways to sort of ensure that plasmid free cells do not grow is to add a lot of antibiotic in the fermenter. There are issues with that which we will talk about in the next class. Why we cannot add too much of antibiotic. We can add antibiotic on a plate, in a small plate, while selecting for new colonies or new clones. But to add them in large scale fermenters has various issues, including environmental issues, which you are very familiar with.
Yes, sir.
Okay, thank you very much.
Thank you, sir.
Thank you, sir.
Right. Thank you.
Sir, one thing I want to ask.
Yes.
Sir, what about the books for this course?
हाँ, वो आपने बोला है और मेरे दिमाग में है. I was very much in my mind. I will have to locate books. I could not locate any recombinant DNA book because I had located books on enzyme, on enzyme catalysts, because that is what I thought I would begin with. So ये वाला section के लिए आपको मुझे time देना पड़ेगा थोड़ा. I mean, next week I will definitely arrange for something. Okay? Because these things are not available easily in books. Yeah. Okay. Thank you.